Hello, everyone. Welcome to Agribusiness Global Live. My name is Renee Targus. I'm the editor for Agribusiness Global and your host for today's webinar, New Formulation Trends. We've got some great panelists and a great discussion that's about to happen. I've got a couple of events that I wanted to tell you about. We've got the Agribusiness Global Trade Summit happening on August 5th and 6th. That will be in Orlando, and it's a great event. A lot of networking and business deals being done. We still have the early registration rate, so I hope that you will check that out and join us there. Also, on April 23rd, we have a Agribusiness Global LATAM virtual seminar. It's free, and it'll be presented in Portuguese, Spanish, and English. But we also have a translation on the presentation. You'll be able to get it in 30 different languages, but you have to actually attend the event live. So that's free, April 23rd, focused on the LATAM region. We hope to see you there. All right, we have a lot to discuss today, so I'm going to do a brief introduction of our panelists, and then we are going to just dive right into the conversation. So on our panel today, we've got Shaun Selness, Head of Analytical Sciences, Small Molecules for Bayer Crop Science. We've got Ashish Batra, Vice President of Crop Health R&D for Corteva. Sara Monteiro, Head of Global R&D for Biocontrol and Adjuvants for Rovensa Next. Leslie Schmidt, Global Account Manager Agro for Evonik. And Joshua Colmer, Founder and CEO for TraitSeq. Welcome, everyone. All right, Shaun, we are going to start with you. We're just going to dive right in and, with all that's going on with banning of chemicals and sustainability, with Bayer's creation of new active ingredients, what focus areas are being considered?
Okay, so thanks, Renee. So a couple of things I want to talk about. Two new active ingredients that we expect to be on the market in the next 1 to 3 years, but then really talk about our discovery process, our crop key approach to discovering new molecules, and really reiterate our commitment to discovering new innovations in the crop science space. As many of the folks on this call know, resistance is always coming, and so really trying to focus on new modes of action, providing those growers with the right tools at the right time to combat resistance across a multitude of pests. So the two active ingredients that we're very excited about that are coming up in the next couple of years, there's a Plenexos, which is our new keto insecticide, which we expect to launch in the next year. In Brazil, really hitting those sucking pests. As we know, Brazil is oftentimes a very challenging environment to control insects, and very excited to be able to introduce that into that market. It's expected to be applied both via soil as well as then foliar. So it's actually the first of its class to be able to do that. So very much excited. It cuts across soybeans, cotton, fruit and veg, and really will provide those growers with a new tool in their toolbox. And then within the next several years after that, we expect Ecofolin, which is our new post-emergent herbicide, which is actually a new mode of action in that space that the industry hasn't seen in multiple decades. So very excited about being able to provide that to growers also within the next several years. Really absolute fantastic safety profile. And that kind of leads into then the crop key piece, because Ecofolin itself, we would call kind of our first generation crop key. It was really a profile that was driven by the early safety profile, not just the efficacy of the molecule itself. So we really focused on designing the molecule, obviously for the efficacy, but really keeping safety in mind very early in the process. So kind of represented our first generation, and really then pivoting into our next generation or iteration of crop key, where we're leveraging digital tools across all those elements. So efficacy, the biology, even up to the target selection itself. So not just optimizing the chemistry, but really picking the right mode of action, and then really designing the right molecule against that particular mode of action. So think of finding the right lock and then finding the right key to unlock that lock that really does it in a specific way. So it really favors then the safety profile of the molecules that we discover in advance, but really then taking into consideration things like safety very early in that process to really maximize the potential for that molecule and the regulatory probability success down the line.
Yeah, definitely.
Yep.
What about is there a consideration going into integrated pest management when creating AIs and how they'll interact with biological products?
Yeah, so definitely integrated pest management is really at the heart of all the technology platforms that we have within Bayer Crop Sciences, not just synthetics and biologicals, but specifically for synthetics and biologicals, we try to design our formulations and our products to be compatible with what we would expect to be used in that market. And then from a biologicals perspective, we also design our biologicals to be compatible with, the expected synthetics that are on the market. So a good example of that is Plenexos. So we designed that formulation to be compatible with a number of different products to really then give that complete toolbox, if you will, to the grower.
What about drone sprayers and other ag tech, as a consideration when you're formulating?
Yep. So, drone technology and then targeted application, which is kind of a broader category, definitely a consideration when we're developing our products. Certainly we're targeting certain markets, and some of those markets are using drone application or see and spray type of technology, and that does require sometimes a different approach when developing the product. But certainly, when we're early in the assessment of a particular active or compound, those are considerations that come into play. Specifically, an example would be, Red Tizo has, technology, basically drone technology that now is partnered with our FieldView, technology to then really allow the grower to best position the product on the right hectare or acre in their farming practices. And we certainly take that into consideration at all stages of development of a particular compound and formulation.
Okay. All right. So, registration has always been a big issue, obviously, in the industry. What's your company's experience been in dealing with that for your new products?
Right. So, and from a global perspective, it's an ever-changing landscape. So we've definitely had to adjust and make sure that we are meeting the highest safety standards wherever we launch products. It has been a challenge, but really leveraging our historical data and our understanding of the regulations and the safety requirements that come out of that, we've been able to really adjust how we've tested our products early, as well as through the late phases to make sure we hit the highest level of safety standards. We hold all of our products to a very high level of safety when it comes to launching products. It is challenging in an evolving environment. Regulations aren't always, synchronized across geographies, but we do try to be very much mindful of those changes and try to anticipate them so that the molecules that we discover are truly future ready.
Great, great. All right. Well, thank you, Shaun. So we're going to keep moving on with our questions. And Ashish, I'm going to be asking you similar questions, to kind of what we talked about. So Corteva's creation of formulations, what is your company considering when you're creating these?
Yeah, so specifically with formulations, more and more premixes. If I look at the last 5 years or so, more than 75% of the formulations that we launch roughly 150 odd new products every year. They're premixes. At least 40 to 45% of them now have at least 3 or more active ingredients. So that's certainly a key trend that we are focused on. Trying to have built-in adjuvants, for example, that enable you to ensure you have the right spreading, you have the right retention, you have the right penetration, you have the right uptake through the plants. So you're able to fully optimize the efficacy potential of the active ingredient. So those are maybe two key themes that we really focus on. Obviously, we're an industry leader in natural products. With natural products come there, again, unique challenges with respect to how you formulate them, how do you ensure that there is compatibility with everything else that's going to go into the tank. So to just give you a couple of examples, we just recently launched Reservoir Rev, which is a ZC formulation, which is essentially a capsule suspension plus suspension concentrate combined together. What it enables is a broader window of application, 3 different modes of actions coming into play. So the complexity of formulations continues to go up as you're trying to bring many differentiated modes of action to manage resistance all in one application to provide farmer that convenience and that really choice, so to speak. So that's really the key emerging area for us.
Okay, that kind of answers my second question about the integrated pest management and, biologicals and, synthetics working together. Is this a trend that you've seen change over the last, decade? Before there used to be a lot of camps between synthetic and biologicals, but now it seems like everybody's kind of focused on it together. Is that definitely a new trend?
I would say certainly over the last decade or so, there's been more of a shift there. But when you think about integrated pest management, in my mind, for us, it starts in the seed, then on the seed, and in the field. Right? So what do I mean by that? So in the seed with the genetics, the germplasm, plus the trait packages that we bring forward. So when we think about herbicide tolerance, so Enlist is a great example for us, which is on soybeans on greater than 60% of the acres, wherein you're bringing forward a chemistry solution, which is industry leading in terms of its risk reduction potential. That's one example of the combination of in the seed and the chemistry. Then on top of it, you have products like Nutricia N, which is a highly innovative nitrogen fixing solution. So thinking through formulation challenges of what all else is going to be in the tank and ensuring that there is compatibility across all of these solutions. But for us, it goes across the entire spectrum of what's on the seed as well. So when you think about seed applied technologies and flowable concentrates, we have now examples of wherein there might be up to 6 active ingredients in that flowable concentrate. So that's in some respects a dream for a formulation chemist to be able to formulate something that has up to 6 active ingredients, some of which may be an insecticide, fungicide, nematicide, inoculant, polymers, binders, all of these coming together in order to enable that integrated pest management solution.
So I have a follow-up question for Shaun and for Ashish. And so Ashish, will start with you, and then Shaun, I'd like you to weigh in on it. Do you think that with all of this money being put into regenerative agriculture and sustainability, do you think that the synthetic do you think the biological will ever kind of surpass the synthetic in formulations? Or do you think that synthetic will just there'll be more formulated to, not harm the soil or, along those lines? Ashish, what's your take on that?
Yeah, so I think it's first important to define what do we mean by regenerative ag, right? And it's really comes down to focus on outcomes. What are the outcomes you're trying to drive? First and foremost, being productivity. You want to be able to deliver more with the same land that is in production and not add additional land into production. So sustainable chemistry can continue to be a very powerful solution to enabling that. So when we think about the sustainability criteria that we put in place for our discovery and development of synthetic chemistry, that includes novel underexploited modes of action, low use rates, less environmental persistence, less risk to groundwater, all of those factors, which are also enabled by biologicals. With biologicals, there are current challenges today in terms of the amount of efficacy that you can deliver. This is where we are leaning really hard into our legacy with natural products and gene editing, and being able to come up with solutions potentially into the next decade where you can have next generation biologicals that do deliver CP level efficacy, CP level consistency, CP level formulation stability. All the right tools are getting together in terms of machine learning, in terms of formulation stabilization technologies that can enable that. But For us, it's more the complementarity between CP and biological, not necessarily at this versus that scenario to enable those regenerative ag outcomes that we talked about.
Okay. Shaun, what's your take?
Yeah, so certainly to echo and rebound on Ashish's comments, regenerative ag and sustainability really require that we think about the system in whole, right? So the seeds, the chemistry on the seed, coming over the top of the crop, and really thinking about where we best place those products for the maximal benefit. So the productivity that Ashish mentioned. So I don't see it necessarily reducing the need. It might change the profile of the compound that we have to deliver in the future. But if you think about the ever-growing, pest spectrum and the changing pest spectrum, I think we're always going to have to require, tools from all walks of life, if you will, to really effectively manage those hectares and acres for the grower, and making sure that we put them where they really need to be, and that, as you indicated before, that when we do put them there, it does what it needs to do, and then really minimizes that impact. A great example of a technology that's been around for a while, obviously, are the herbicide tolerant traits that enable things like no-till, which then minimizes the release of CO2 in the air. So those are all technologies that have been around for a while, but we want to continue to build on those and bring all the tools to bear on the challenges that the growers face, but also that the planet faces.
Okay, great. Thanks, Shaun.
Yep.
Ashish, I've got one more question for you, and then we're going to be going to Sara to talk a little bit more about biologicals and plant health. What about drone formulation and ag tech as far as creating that?
Yeah. So with drone applications, certainly from a formulation perspective, you want to worry about ultra low volume applications, water volume applications. So that certainly poses some unique challenges for a formulation chemist. But also when you think about launching a formulation like that, you want to put in enough versatility that if a grower out there wants to use a backpack sprayer or a conventional sprayer, that it still works with that, right? So a great example for us has been a formulation we launched in APAC for the rice market, Hibifen, which brings Pyraxol, which is a new differentiated mode of action for brown plant hopper, combined with Gemvelva, which is again a natural inspired product for us. And the way the formulation is designed, it works both from a drone application perspective, but also a backpack sprayer. So there is that versatility built in. But the other thing that you have to also start to consider is drift. With the number of drone models that are out there, and each of them with different number of sprayers, the position of that sprayer relative to the rotor, you have to be able to do the right modeling and the right experimentation to make sure, A, you're having the appropriate swath coverage that you want, you're ensuring that you're not going to inadvertently create more of a drift challenge when you're using these drone applications. So those are two very specific areas we focus on in designing products for drone spray applications, versatility for it to be used both in a drone and outside of a drone, and being always very, very conscious about drift implications. APAC is certainly a growing market. Brazil, there's tremendous adoption going on. So all the key geographies where we try to enable this.
Great, thank you very much. All right, well, we're gonna move on to Sara and talk a little bit more about biological formulations. So Sara, with Rovensa Next, they've got laboratories all over the world. What trends are you seeing in the formulation of biologicals and plant health?
First, thank you very much, René, for the invitation to be here in this very interesting discussion. And to begin with the biologicals, just want to clarify that I'm going to talk about biologicals, but not only on microbes, so living cells for us in Rovensa. Biologicals mean everything that it's a microbe alive, but also some metabolites from the microbes and some natural plant extracts. So, and this is where I'm going to focus my conversation today. Because formulation of microbes or live cells, the challenges are slightly different from other type of biological solutions. So we cannot, the challenges that we are facing are not exactly the same. But nevertheless, you are very correct. Because I have labs in Rovensa and X has labs in all the geographies, so we face different challenges regarding the needs of the growers in each of the geographies. So we cannot develop the same type of formulation of even the same type of microbial in Brazil that we are developing in Europe, for example. Because for us, the first target on developing a new product is to address the needs of the growers. To answer the needs of a particular disease, of a particular pest that the grower has in that particular region. And to do that, what we are doing and what we think that is very important is to understand the needs and to develop like more or less tailor-made solutions for each of the geographies in order to answer the real needs. In terms of formulations development there, yes, we are also facing, as already Shaun mentioned, the regulatory pressures and the regulations in each of these geographies are slightly different. And because of that, we have, our tools are different depending on geographies. Of course, that we at Rovensa and X, we are always looking for the most sustainable and biological, so even for our co-formulants in the formulations development. But indeed, we have different opportunities depending on the region that we are developing the products. And yes, it's more or less regarding the geography landscape that we have that I can say in terms of our developing of products.
So, for the I asked Shaun and Ashish about this. Are you developing biologicals that will be able to go along with synthetic products, or are you just targeting, I know nematodes in Brazil are a big issue because they had a synthetic chemical that was banned, and so that became an issue. Is that something where you're trying to focus your products on?
For sure, and I think the biologicals are now out there for a long time. So I think we all learn what the biologicals can do and what the biologicals cannot do, and how the biological also needs to evolve in order to answer the growers' needs. And we know that the biologicals need to work as a tool on the farmer selection of opportunities. And of course that the syntheticals will also be there. So we need our biological tools needs to be able to handle also the presence of other compounds in nature. And this is why today we at Rovensa and X, we are in terms of evolution, we are not just looking at the microbe of the cell, the microbe alive, but also looking at other opportunities like metabolites from the microbes, the plant extracts or products from the plants, and also some fermentation products. We are also looking at RNA technologies, for example, proteins technology. So molecules that are natural, that come from these natural sources, but they are more fit maybe in these counterparts chemical products. And this is where we are going. Today, our goals is not just formulating a microbe as a standalone solution, but work in combinations and to give some, more resilience even to the microbial cells. And this is what we are doing. So we are developing our products, for example, using structured liquids in order to give more shelf life to the products and to give a better formulation type for the grower to use more or less like they are used to work with the chemical products. And also we are trying to do combinations between our alive cells and also, for example, with some metabolites, but of course at protecting the microbes to these other types of chemistries, but to give them resilience and more persistence in the environment and to be able to handle with this tank mix that are the common way that the grower is using today, all the tools.
Okay, that's great. So that's kind of along the lines of where you see the future heading with biologicals as well as kind of just what you discussed, or do you see other trends happening that you guys aren't actually doing yet?
No, for us, I think this is the future of the biologicals. So we should not consider the biologicals only as the living cells, but also as combination with the other chemistries and trying to formulate even the biological cells in order to protect them, to be able to resist to the environment. Because, you know, in the lab conditions, it's very easy to have a biological perform really well on diseases or even or with insects. But when we arrive to the field conditions, when the microbes need to survive UV radiations, when they need to survive, for example, the application tools, the spray application tools with very high mechanical forces, the cells are very susceptible. And this is what we need to think. We need to give the tools, or we need to give capabilities for the microbes to resist to these real field conditions. And this is our main, main formulations development today. We are, of course, looking to new active ingredients, new modes of actions. But in addition to that, we are also looking at application methods, at giving resilience to these biological tools to be used. What I want to say is that the grower can use this biological in the same way that they are using the chemical or the synthetic tools, to not complicate and to make the biological tools much more user-friendly to the growers. And for me, this is the future of the biological, is to create, biologicals will be here because they are a very important tool for restoration and for the soils and for our environment. We are all facing these climate changes. So microbes are the key for these challenges that we are facing today. But we need to make these biological tools also user-friendly for the growers. So I think this will go in parallel. New tools, but also making these new tools user-friendly.
I think that's exactly it. I mean, that's one of the biggest pain points for a lot of the growers, that they say is that, they're used to doing a certain process, and if you've got a product that is coming in, or it can't be applied like they normally do, then that decreases adoption. So all right, I'm going to move over to Leslie. Thank you, Sara. And Leslie, you were talking a lot about drone formulations. Sara was just talking about, spray equipment, and we talked about with Shaun and Ashish. So what's going on with the adjuvants and inerts as far as that goes along with the...
Yeah, I think especially when you look at adjuvants or surfactants for drone applications, there's a couple things that have already been pointed out in today's discussion, but I'll talk a little bit more about. There's a lot of opportunity with drones, right? We saw really the industry pushing from APAC. We've already got active formulations out in that region, but we see it across now coming into the Americas as well, and also into Europe. There's tons of opportunity, but there's a lot of challenges. And really this comes from the massive reduction in water volume that you have. And so when you're looking at a formulation in general, surfactants historically were used in tank mix, right? So you had tons of water space to get your product diluted. We saw over the last decade or so that a lot more built-in adjuvants. So we're going from being just in the tank to now being in the formulation itself. So we've already have a little bit of a reduction of volume. And now when we go to drones, it's even heightened more. And so when you have that reduction in volume, you have a lot less space for built-in adjuvancy. And we need to make sure that the adjuvants really are the right ones for that application. The other thing that you see is there's different nozzle types. So your droplet sizes, the droplet speed is going to be different. The height of the spray is also going to be different. And the force of those droplets hitting the leaf surfaces is different. And so you're going to have the way that adjuvants typically are used, where they help adhere to the leaf surface, they help with penetration, they help with spread. All of these things can change just by putting the same chemistry into a new application. So those are where we see most of our research and also the industry looking at how do we best use adjuvants and surfactants in these applications.
Great, thank you. What about the drift reduction? I know CPDA is working hard to, about the Endangered Species Act in the US. What sort of trends do you see going along with that?
Yeah, I don't see drift as a new topic. Drift is something that both the crop protection and the adjuvant companies have been trying to solve for decades, right? It's nothing new. But where we do see new requests is that people are looking for some of the solutions to be a little bit more sustainable. So when you look at the industry in general, we may talk a little bit more about sustainability, but in the drift particularly, making sure that the solutions are not microplastics is a big topic. So some of the chemistry can be potentially defined that way. What we look at for drift reduction is there's a very specific definition. If you're going to say something is a drift reducing agent, right, you have to have a certain particle size target. And so when we look at how do we use adjuvants, we look at not just whether or not we can meet those targets, but how much can we actually reduce the amount of drift. And so what I see is potentially using different chemistries together to meet those targets.
What about drone formulation? I know that's been a subject that we've talked with experts, for several years, and they keep talking about like this is an area where the market there's opportunity. What's your take on that?
For drones, yeah, we spoke about it just a minute ago, but again, drones are a big opportunity, but they have challenges as well. So I think what we are doing is trying to partner with our customers as much as possible, not just on the crop protection side, but also the companies that are actually designing the drones themselves. How are they looking at designing the equipment? How much space is there, not just for the crop protection product, but for an adjuvant to exist in the application as well? It's also really interesting to see that in different regions, the requirements and the styles of drones are very different. And so I don't see that there's going to be a one size fits all type of solution in this area. I think that there really has to be sort of a different solutions and not just that our customers, our crop protection formulators, but ourselves as adjuvant designers need to really understand how the different equipment is going to, how our adjuvants are going to behave in the different equipment.
Perfect. Thank you. Thank you. All right. So I'm going to go to Joshua because we're kind of getting a little low on time. But Leslie, that was really interesting. And I do have some follow-up questions for you. But we do have 14 questions from our audience right now. So let's talk with Josh and then we'll get to our audience questions. All right. Joshua, TraitSeq is working with Syngenta to formulate biostimulants and analyzing research data, specifically biomarkers. Can you give a comparison of how TraitSeq is speeding up the analysis in comparison to how it would be without artificial intelligence?
Yeah, absolutely. So traditional research involves selecting biostimulants or crop protection products that have activity in the lab or the glasshouse, and then assessing their activity in the field. But unfortunately, as Sara was saying earlier, depending on the type of biological, the correlation between the performance in the glasshouse or the performance in the lab with the performance in the field is not always very good. And this can be because there are complex interactions between the biological, the plant, and environmental factors that haven't been analyzed in sufficient depth beyond just looking in the glasshouse or the lab at the phenotypic effects of the compound. But if one leverages AI and machine learning, it can be possible to gain a significantly better understanding of what controls the activity in the field and at a molecular level, how the biological can affect important agronomic traits through identifying biomarkers like we will be doing in the partnership with Syngenta. And then following this, it's also possible to use AI or machine learning to then identify the most promising candidates, so the ones that are most likely to be active and effective in the field. And this is broadly what TraitSeq brings to the discovery and development process. So using AI in two different ways. So firstly, for biological inference to better understand at the molecular level how these candidate biologicals work and interact with plant genetics and the environment. And then the second application being for predictive purposes to optimize the selection process for advancing candidates through the development process. And then just to touch on the impact of this, given in that some cases you can only have one attempt at field trialing in a year, this sort of optimization could shortcut decision making by literally years. meaning that products could be commercialized earlier, and ultimately the farmers and growers get access to well understood, effective, and more sustainable products earlier.
Great, thank you. So what's your take? Do you see quantum computing becoming a part of analyzing research and development in the coming decade? And if so, how would this affect formulations?
Yes, I think quantum computing and other data processing and data analysis areas are very exciting, in that they can allow us to model and analyze plant input environment interactions at a greater scale and at hopefully unprecedented levels of detail, and even faster rates, meaning that more options and formulations can be considered, and that would result in a better chance of these products being selected for field testing and being candidates for commercialization.
So I'm gonna, one of the questions I wanted to ask you about, this data is, do you think that this could be incorporated into registration processes, and would that be a possibility of speeding them up?
Yeah, so I think probably people like Shaun and Ashish are more of the experts on registration, but from my understanding, the process is quite different across different geographies, and there's sort of a bottleneck in the number of people that are assigned to process the submissions, and the fact that not all of the regulatory packages that are submitted are fully complete means that there probably is an opportunity to exploit machine learning and AI to process and make decisions, as there's going to be an increasingly large amount of data coming in. So yeah, I think that there is an opportunity to use developments in AI to assist in making decisions about registration applications and reducing that bottleneck.
We hope, or that registration bottleneck is like the pain in this industry. All right, we've got 17 questions from our audience right now, so I'm going to dive into that so that we can try to get as many of these as answered as possible. The first question we have is regarding CS formulation that uses microplastics and will not be able to use it anymore in the European Union. What suggestions do you propose? Who'd like to take that question?
I can maybe take a shot at it. I think the question is spot on that given some of the microplastic regulations in EU, there are a lot of companies that have been focused on coming up with biodegradable solutions, right? A lot of startup ecosystem has focused a lot of energy on it. Some work, some do not work. Some work to a particular scale, only at the lab scale. But when you're talking about scaling up to the thousands of liters, scale-up challenges emerge. So certainly there is a vast ecosystem out there that is trying to resolve this challenge, all the way from small startups to academic institutions. So at least from our perspective, we tend to sort of be tapped into that entire ecosystem to continue to look for the best possible solutions that, again, vary from what exactly is it that you're trying to stabilize. Is it a small molecule? Is it RNAi? Is it peptides? Is it proteins? What is it that you're trying to encapsulate? So this entire area of sort of formulation stabilization technologies, and I throw in capsule suspensions in there primarily because capsule suspension is the classical way to stabilize an active ingredient. A lot of work going on, but no one size fit all solution. It is very much dependent upon the specific active ingredient, either biological or small molecule.
Okay, great. Anybody else, Shaun or Leslie, do you guys have anything else to add to that?
Yeah, I think, really thinking about the performance of the product, making sure that, if we're considering substitutions, that we really don't compromise on that, because at the end of the day, we need to really make sure that the product continues to perform in the field. So we continue to look at those alternatives, but as Ashish said, that it doesn't always scale, but we are certainly connected to the vendors and the providers in that space as we continue to develop, future ready formulations.
Okay. Sara or Leslie, did you have anything else to add to that?
Yeah, the only thing I can add is from an adjuvant standpoint, what we do is also look at alternate formulation types, and I'm sure that all the companies are doing this, right? Why is the active ingredient need to be encapsulated, and is there another way that it can be delivered? And so with the all of the regulation around microplastics, this is just a really challenging area. So if there's a different way to deliver the active ingredient to the plant, then that's what we will try to formulate for.
All right. Sara, did you have anything you wanted to say, or?
Yes, it's more or less for us. So microplastics are completely out of our business, eh? And but yeah, we are. And for the case of the microorganisms, we are trying to do encapsulation, of course, and trying to isolate but with, of course, with alginate solutions, chitosan-based solutions, in order to be, very sustainable. But in order to be able to design formulations, stronger formulations in can, but with the microbe isolated to not contact or to keep the stability of the microbe, but having the other possibilities for formulation development in order to give more spreading and wettability and dispersion of the microbes as soon as they reach the target.
Great. All right. Thank you. All right. Next question. Would you can you give some insights into carriers and excipients used to make synthetics and biologicals compatible? Does anybody want to take that one?
Yeah, so I can start maybe, because this is an area that Evonik has done quite a bit of work. And what we have found is that there are some, I would say, carrier fluids that you can use that are very good at stabilizing microbes. And what we found is they're also very good at stabilizing active ingredients that are a little bit water sensitive. So typically you would have to go to a dry formulation or an oil formulation. We have found some other solutions. These come with also, I would say, unique formulation challenges. So you still need to have a formulation that is physically stable and easy to use in the tank mix. And so I would say there are some options out there, but sometimes it does take a little bit different formulation technique to make them completely successful.
Anybody else want to add anything to that?
Yes, as I was referring when I spoke, for example, one opportunity is to do like a structured liquid that you can separate. So you will create a liquid formulation, but with a solid and spatial positioning of the molecules. So in that case, you can have a compatibility, because the microbe or the biological will not contact with the other parts of the formulation, and we can have like not an oil formulation, not a powder, but something in between that will give stability, that will give shelf life, and that will give compatibility with some other chemical products.
Thank you. Ashish, were you about to say something?
I was just going to say that, this is where maybe leaning into what Josh was talking about, bigger data sets, high throughput experimentation, being able to do a lot of screening, and particularly when you're talking about microbials, doing high throughput enumeration assays and starting to build that data set that you can then eventually tap into machine learning models to start to make some predictions. I think that's an area that is still somewhat nascent in the industry, but something that we need to continue to drive towards, because very rarely will be there a magic ingredient that works across the board. So classes of chemistry, classes of solutions that work in some cases, but you need to start to build those data sets, and that's where high throughput experimentation and screening along with machine learning tools play a very critical role.
Great. Josh, did you want to add anything to that, or?
Yeah, I was just going to say, I think that despite all of the advances in machine learning algorithms, something that I think that is incredibly important for the success of any project or experiment now and will continue to be in the future is the quality of the data set, especially when working with omics data that's tissue sensitive and time sensitive. All of these factors need to be considered to sort of maximize the signal to noise ratio in the data set that you're generating. So I think that it's exciting that the advancements in machine learning have come at a similar time as when the capabilities of companies in our industry have increased in generating labeled phenotypic and molecular data sets.
Great, thank you. All right, Shaun, we've got a question for you. Considering that active ingredients are contributing to most of the safety concerns in formulations, how broad is the tolerance for co-formulants having certain level of labeling?
Okay, need to maybe better understand the question, but if I understand correctly, there's the comment around safety of the active, but then in combination with either surfactants or other formulation ingredients. Certainly, as I iterated before, we're really rigorously test obviously the safety of the molecules coming forward. So we really make sure that the molecules coming forward do not have major concerns that limit their utility, but then also taking into consideration what we're combining them with from a formulations perspective. I think it was said earlier that with the different kind of requirements and the different regulatory bodies, really making sure that we're formulating with ingredients that can give us the broadest impact while making sure that the safety of that particular product meets regulatory requirements all across the way. But it's certainly something that we test very rigorously.
Okay, great. Next question. Would you say that the movement toward the use of MPF polymers is a reality around the world in large pesticide and seed companies? Anybody want to take that one? No. All right, we'll have to get back to that question at a later time. What shelf life, and next question, what shelf life in general is expected for biologicals? Will it be possible to achieve 2 years shelf life for biologicals, particularly for proteins and peptide bios? Sara, did you want to talk about that?
Well, again, when we talk about biologicals, even inside of the living microorganisms, we have different type of cells. We have the gram-positive and the gram-negative, and they are completely different in terms of stability. But to answer the question, yes, it's possible to have 2 years storage stability of some of the biologicals. Of course, even in the case of the proteins, and which is the question, proteins, if they are well formulated and if they are, for example, solid formulations, powder formulations in the case of the biologicals are a very good way to keep the stability for 2 years, close to 2 years. But even so, even in liquid formulations, yes, it's possible. With the current technologies, it's possible. And there are several products we have. So Rovensa Next has some products even based on microbial solutions with more than 2 years of stability. So that is possible.
All right, great, thank you.
Yeah, Renee, maybe just to maybe add on to that, one thing to consider there, we're not necessarily just talking about stability in the jug or in the formulation, but certainly when it's on the seed. So those are all aspects that have to be taken into consideration. But as Sara said, I think we have approaches to be able to do that in multiple environments.
Great. Okay. Ashish, this question is for you. We know the grower will tank mix with various other products with Enlist. How do new products get listed on the Enlist approved tanks mix partner sheet? And is there compatibility testing done internally at Corteva, or does a company need to show proof of compatibility and stability? And is there a working group we can submit to?
Sure. So through the product development process, we spend a lot of time doing internal testing of any new product that we are bringing forward to the market with what we know to be the most common tank mixes that a farmer is going to use, whether it's fungicides, insecticides, herbicides, whatever the case may be. But obviously, you can't account for every possibility that might exist. So we tend to look at probably the most commonly used. It's specifically for Enlist. I think that number is in the hundreds that have been tested already internally. So there isn't necessarily a working group that you submit to. It's more, what is the farmer likely going to be using? That's what we typically tend to test for. But in terms of, if there is one esoteric formulation that somebody's developed, then I would say that's incumbent upon them to do the testing. Versus from a product stewardship perspective, we tend to focus on the vast majority of the market that is going to be time mixing, say the known 100, 200 formulated products that are out there. So that's how the process works.
Okay. Ashish, there's another question for you.
Yep.
They were talking about the same formulation you said is used in drone and backpack sprayers. Is there any change in the dosage?
Yeah, I mean, eventually it comes down to grams of AI per hectare that you want a final application to look like, right? So it depends upon the water volume that's going to be in the drone versus the backpack sprayer. You're going to adjust that to deliver the final active ingredient per hectare that you want to get delivered.
Sara, this is a question for you. Can you tell us more about the use of structured liquids for biologicals?
Yes, okay. So, structured liquid for biological means that we are not using oil formulations. Today, for a liquid formulation, a oil dispersion is the medium that it's a medium free of water. That means that it's the adequate medium to favor storage stability of the spores. But besides of these vegetable oils, we can create like a structured liquid based on water miscible polar liquid carriers, for example, that are very good to keep stability. And in addition to that, this polar media, the selection of a polar medium, that means instead of oil, so a non-polar, it will make an easier solution on when the product arrives to the field, because it's increased the dilution of the product without the need of the growers to use like any type of emulsifying system in the tank. So it's to create like a structure, like a jelly structure, where the microbial solution is more or less encapsulated or more or less stabilized in this jelly solution, but it's in the end is a water-soluble solution.
Okay, that kind of leads to the next question, and this is again for you, Sara. It says, in the case of biologicals, like entomopathogenic fungi, maintaining CFU is very difficult in field conditions, as told by Sara. Are gel-based formulations able to entrap a larger number of counts? Is it possible?
Yes, it's possible. And I think we can go up to 10 to the 13 CFUs per liter of product with this type of technology. So yes, 10 to the 9 per milliliter, or 10 to the 9 in case of grams in this type of solutions. Yes, so it's possible.
Okay, great. Leslie, this is a question for you. It talks about drones and the drift problem, is significant. Can we use electrostatic type nozzles to bind to plant surfaces better? What's your take on that?
Yeah, I'm not a nozzle expert, so I'm not going to be able to answer that question directly. All I can tell you is that with the drones, there are many different nozzle types. And what we see is when you typically measuring for drift, you're using like a flat cone nozzle, right? And once you get into drones and you get into the rotation of these nozzles, everything that we know about how to measure droplet size kind of goes out the window. And so there is a lot of experimentation. You really need to know, again, what the equipment that you're looking at, what the nozzle type is, things like the height of where you're going to be spraying. But ultimately, it is going to come down to being able to test in that individual system.
Okay. The next question is kind of, I think we've answered it before, but it's phrased a little differently. So I'd like to ask it. It's talking about how formulation is changing to mix biological and chemicals into the same products. Are there new formulation technologies being developed for slow release active ingredient products? And Leslie, would you like to answer that one, or Shaun?
Sure, I can start. I'm sure other people will be able to fill in. But we have been looking at mixing biologicals and chemicals together. And so we do have some products in our portfolio that we've seen some success and even been able to show mixing a chemical active ingredient and a microbial in the same formulation is possible. But it's not easy, and it's going to come down to the individual active ingredients. If you have a fungal active ingredient and you have a fungicide, obviously putting those together into a formulation is very challenging. So you really have to think about the full concept of what you're trying to put together, and how that's going to be applied. And again, how much space you have in the formulation. So I think that when you're looking at how to put these formulations together, there are options that are available, but they make your formulation much more challenging to get to the market and to become stable. And so really thinking about what is the best formulation type for that combination of actives, or does it make more sense to have them as two separate products, is something that you have to do as a as a formulation designer, and making sure that you're getting the efficacy that you need without giving yourself too many challenges in one can.
Okay.
Yeah, maybe to add to that, certainly, we try to avoid solving too many problems with one solution. That oftentimes gets us into challenging spots from a formulations perspective. But another aspect to consider is, early on in the selection of the active or the biological, is thinking about its compatibility in a formulation and how you might be able to deliver that. So bringing that earlier into the design of the actual active principle is something definitely that we consider. It's not just the formulation, it's also then the active that might dictate the formulation and the strategy we might take there.
Okay, great. Anyone else have anything to add to that?
Maybe the only thing I'd add is that when we talk about biologicals, I think specificity matters. What biological are you talking about? Are you talking about a living microbe? Are you talking about a plant extract? Are you talking about a plant growth regulator? Are you talking about copper? I mean, the term biological gets thrown around, but it's very critical for us to start getting very, very specific, because each of them has then a very particular nuance. So if I'm thinking about a plant growth regulator, which is almost like a synthetic equivalent of something that already occurs, the challenges there mixing with the synthetic chemistry are fairly limited compared to when you're talking about, say, a peptide or an RNAi or a living microbe versus even a plant extract. So each of these results in a very different, unique solution. So just wanted to sort of emphasize that when we're talking about biologicals, it's critical that we get really, really specific as to what type of biological are we talking about.
Great. This will be our last question, and Sara, it's for you. And then we've got about 30 questions, and we'll be answering those in a blog. So if you didn't get your question answered today, we will get to that. You will have your question answered eventually. So Sara, drift versus volatility. Do you have any insights into designing formulation to combat drift plus volatility?
Ooh, this is a very interesting question and very difficult to answer. When you are talking about drift, you are talking mainly about the size, the droplet size, and the way that the drop reaches the crop. When we are talking about volatility, we are talking about the active ingredient. Yes, you can have, it's a completely different behavior. If you have a volatile active ingredient, you need to protect the active ingredient by encapsulation, for example, by slow release of this active ingredient, and only after we can consider the drift. So the drift is more related in the case of a formulation development. Volatilization is more related with the active ingredient. So I think I don't have a straight answer for this. It's a difficult subject.
Yeah,
indeed.
it sounds like it. So, but thank you for giving what you did. So I appreciate that. We have run out of time, and I know we have a lot of questions that were not answered, but we will answer those in a blog along with the recording of this. You'll get that in about 2 weeks' time. But I just wanted to take some time to thank all of our panelists for joining us, and thanking you for joining us in this webinar. It was a great discussion, and we will be following up again with the blog and a recording of this. So at that, then we'll be ending this, and I just want to thank you all again for coming, and we hope to see you again at the Agribusiness Global Trade Summit and the virtual event that's coming on April 23rd. So hope you have a great rest of your day or night wherever you're located. All right, thanks everyone.
Thank you very much.
Thank you.
Bye-bye.